If you've listened to Brewlosophy at all, you know how much we love Imperial Yeast. The company was founded on the ideal that if you're going to do something, do it right. From their Imperial Pitch Right pouches, packing a whopping 200 billion cells of healthy and viable yeast in each pouch, to their commitment to commercial customers, guaranteeing 10 of their most popular strains are in stock for orders up to 20 liters, or the shipping is free, Imperial Yeast does it right. No more worrying about whether you need to make a starter or propagate your yeast prior to pitching. Imperial Yeast makes it easy for the home brewer and commercial brewer to obtain direct pitches at proper rates when you need it. To place a commercial order or to get more information about everything that Imperial Yeast has to offer, head on over to imperialyeast.com. Welcome to the Brew Lab.
Beer is a beverage best consumed fresh, but sometimes that's not as easy to do as it sounds. For brewers shipping their beers with distributors or transporting them to competitions or over to friends' houses, spoilage microbes, especially if the beer gets warm, can totally destroy the beer that you've worked so hard to make. I'm your host, Cade Job, and today in the lab I'm speaking with Dr. Grzegorz Rachon, who's the Brewing Microbiology section lead at Campden BRI. And over a series of 3 episodes, we've been talking about pasteurization. That's the process of killing off beer spoilage microbes using heat. Apply heat at a certain temperature for a certain amount of time, and you can eliminate any of these unwanted microbes from your beer. So why did we need 3 episodes to say all that? Well, it's because Greg and his team were able to tell a pretty cool story with their research. So they started with a client asking them, okay, well, how long and at what temperature do we need to process packages in order to eliminate microbes from our beer? And it turns out it led them down a little bit of a rabbit hole. The first realization being that there aren't any modern recommendations for beer pasteurization. So that was the subject of episode 71 of the Brew Lab that would talk to the first episode with Greg, where we called it Challenging Beer Pasteurization Assumptions. So then in episode 77, Heat-Resistant Ascospores, we talked about how Greg and his team identified a particularly heat-resistant beer spoilage microbe and how we could use that microbe, yeast ascospores, to set brewing pasteurization requirements. But they noticed something interesting. The hoppy beers in that study required less pasteurization than the light lager, the other beer in the study, which led to the third installment in the series to answer the question, how do hops, or specifically iso-alpha acids, how do they impact beer pasteurization? Maybe hoppy beers don't need as much heat treatment as, say, light lagers. So Greg and his team ran the beer through another experiment, collected their data, and we'll discuss their results and conclusions in this episode. Hey, if you haven't clicked the button yet to become a patron of Brewlosophy at patreon.com/Brewlosophy, please consider helping us out. In addition to the feel-good vibes of helping to support the work we do here at Brewlosophy, you get access to a bunch of awesome rewards depending on the different pledge levels that you choose. You get access to Brewlosophy contributor recipes that we've never published, new discounts each month to YakimaValleyHops.com, and for $3 a month, access to a monthly live Q&A session with a special guest from the brewing industry. This is a really underrated reward in my opinion. Guests like John Palmer, Gordon Strong, Tom Shellhammer, and more are all saved on our private Patreon Facebook page. That means even if you miss them, you can still get access to these at that monthly $3 monthly level. And there's brand new guests every month. So are you kidding me? If you want to up your brewing game, it's a no-brainer to get access to this treasure trove of knowledge. And all you have to do is become a patron by visiting patreon.com/Brewlosophy. Hey, we got a nice bump in reviews in Apple Podcasts after the new year, so thank you so much to the folks that reviewed us. And if you haven't already, here's another call to action for you. And it's also a really easy way to help us out. Leave us a rating or review wherever it is that you listen to podcasts that actually allows you to rate or review. And seriously, I read every review and love hearing from y'all. So yeah, drop a review for the show. I'd really appreciate it. Feedback is brought to you by the imaginative crew at Haas, who developed a revolutionary way to dry hop using Spectrum, a 100% flowable 100% hop-derived product that's fully dispersible in cold side applications for great flavor, efficiency, and less beer loss. No solids means less loss, and it's fully dispersible in cold beer, so there's no contact or residency time required like traditional dry hopping. Spectrum fully disperses immediately, so you don't need to wait 24 to 48 hours or worry about double dry hopping. and you don't have to have any special dry hopping equipment. It stores easily, it's easy to use, saving you precious time and getting instant aroma in each batch. It's currently available to commercial brewers in trial quantities of Citra and Mosaic, so check it out by visiting johnihaas.com. That's John, the letter I, Haas.com. And I just heard from Jeff Daly that there are still spots available for the Hop Flavorist America course offered by Haas in March in Yakima, Washington. It's a 3-day course taught by BrewLab resident sensory scientist Jeff Daly, where you'll learn how to identify and describe the aromas of hops, where those aromas come from, and how to use them effectively. And I just learned that, hey, there's some lunches and dinners included in there too. So if that's an extra perk that you're interested in, go ahead and send Jeff an email at jeff.daily, Dailey, at johnihaas.com for more information. All right, listener Paul wrote in with feedback about episode 94, Competitive Brewing with Jordan Folks. He said, Cade, oh my God, great show. Well, thanks. He said, I really liked hearing the methods of recipe design. The 20-20-20 rule that Jordan mentioned is a game changer. I've landed on something somewhat similar, but this helps out a lot. I don't have enough gear to do the sour thing, but it makes sense if you want to do it. Anyway, I know this isn't the typical show format, but give me more. It's cool to hear the methods of brewing great beer. I've been brewing for quite a while, but haven't invested the time to really nail down the methods to make great beer every time. I also realize it's probably hard to get details of recipe design out of people, but please do it if you can. Keep cranking out the good stuff, Paul. Thanks, Paul. And thank you for the feedback. This is awesome feedback. I mean, again, this feedback is what led us to having Jordan on doing Applying the Science episodes, but, of course, that was a special episode, recognizing his Oshbodhi accomplishment. So yeah, awesome feedback, and I'm glad you like it, and hopefully we'll have some more episodes like this up in the future. So, all right, well, I'll be back in a few minutes with Dr. Greg Rachon, and we'll be talking about how hop alpha acids might reduce pasteurization times.
Established in 1995, More Beer has been consistently serving the greater brewing community since the times IPA was expected to be bitter and clear, and there are reasons they've stuck around so long. In addition to their massive product selection and excellent customer service, More Beer has locations on both the west and the east coast of the United States, which translates into fast shipping times regardless of where you live. And when you spend more than $59, shipping is free. When you're in need of brewing ingredients and gear, there's no better option than morebeer.com, one of the most trusted shops on the planet.
One of the worst things that can happen to your beer after all of the blood, sweat, and tears you put into it is that you go to take a drink of your beautiful concoction and it's become a stinky sock or a phenolic mess. If only you had listened to this podcast and taken some notes about pasteurization. Back with me in the lab today to finish up our 3-part series on heat-resistant yeast ascospores and pasteurization is Dr. Grzegorz Rachon. Greg, welcome back to the Brew Lab.
Cade, thank you very much for having me again. It's a pleasure to share my work in these subjects. I've got a bit of bad news because we're still investigating this, so this may be not the last one. We still may be going on for another 10 years, so I may come back.
Hey, that is fantastic. I would love to have you back, and of course, it is great to have you back on this episode. This is the third part of our series. I won't call it a 3-part series. I'll say it's the third installment so far, and we've been talking a lot about pasteurization and microbial spoilage. It's been a really fun journey, and are you excited to share the next piece of the puzzle with listeners?
Oh, yes. I'm always happy to talk about what we do here at Campden BRI Brewing Research section at Nutfield. Yeah. Super excited. Thank you very much again for inviting me.
Yeah, of course. And just a reminder, so Greg is a brewing microbiology research section lead at Campden BRI over there in the UK. And he's also got a Doctor of Agriculture and Food at the University of Reading and University of Birmingham. And on episode 71, the first episode of this series, he mentioned that this research grew out of questions from Campden BRI clients asking about
pasteurization and developing a more modern understanding of what's going on with this process. But I first reached out to Greg because of this article, the one that we're going to talk about today. It was about hop extracts and their impacts on microbes. And that's one of the reasons why I reached out. And Greg said, well, hey, we've also done these two other episodes or these two other papers. And I think if we, try to talk about all three of these, we can tell this great story about microbiology and pasteurization. And so again, this one, how did you get to this point, Greg? Like looking at the impact of isomerized hop extracts on microbes and spoilage and pasteurization?
Yes, as you mentioned, this work grew from the kind of clients coming to us, approaching us with the specific question, how to pasteurize this product. We've got new product. We don't know how to pasteurize it. It's a medium ABV, for example. We know pH. So we were looking for the specific microorganisms that we can use. Sometimes it's very easy because you know what kind of what microorganisms would be more heat resistant, but sometimes not. And also, if we want to answer this question, we don't want to each time looking for the specific microorganism. We want to have one microorganism that we can use for lots of different products. And looking for this, I find out that there is not the microorganisms like this biological indicator is not validated yet. So we were looking for it. I found out that the guidance are not really up to date. The guidance were quite old, and they were fitting the past, the 30, 40 years behind, but they may not be fitting recent development and what's happened in the brewing industry. And we were conducting first work where we were challenging kind of approach for the pasteurization. We were looking at the spoilage bacteria, and then we showed that how important is to use a specific D and Z value, heat-resistant values for the specific microorganisms, and how important Z value, specific heat-resistant indicator is for calculating pasteurization. And in the second article, we were validating a biological indicator, which we found as a yeast ascospores, and we find out that at the different beers, specifically in the different hop beers, there is a significant impact with correlation of heat and hop resistance. And hop presence, and follow up this second study. We have to find out. We said to ourselves, okay, we have to prove this. Is this really the truth, or this? And anyway, we didn't have enough data, so we had to continue. So this third research came out from the second one as well.
Yeah, exactly. That's a great summary of what we've been doing up to this point. The first episode was talking about updating, like you said, these D and Z values for determining pasteurization, right? The guidance I think we talked about in that show was from like the 1960s or 1950s, right? Something like that. And you were looking at bacteria, and one of the things that you noticed out of that was, hey, these bacteria have these D and Z values, but there's this other, microbe out there, yeast ascospores, that might be more appropriate. So then you do your second round of research saying, okay, well, let's validate yeast ascospores as the most heat resistant or heat tolerant of these microorganisms, so we can have one set of guidelines that we give to our clients, like pasteurize to this D and Z level, and you'll have microbiological stability. And then you sort of looked at that, and you did different beers, right? You test it with different beers, a non-hopped and then a hopped beer, and the hopped beer had less heat resistance. So you say, well, that's really interesting. And then, of course, we get to this episode where we're going to talk about isomerized hop extracts. And so we'll talk a little bit about how and why microbes are impacted by hop acids. We won't go too far into the science on that. But then, we'll do a little refresher on ascospores and where we've been. up to this point, and then ultimately get into the results. Do isomerized hop extracts improve microbial stability? Like if you have hoppier beers, are the microbes easier to, or are hoppier beers, let's say, easier to pasteurize because the microbes maybe are less tolerant? Is that a fair summary of the research and where we're going?
Yes, excellent summary. Sorry, yes.
All right, great. Well, cool. Well, let's start then. I think we should start here. You kind of start here in the paper as well when you're writing the article. And you say it's been well established that hop acids negatively impact microorganisms, right? I mean, I think a lot of times we talk about that was one of the reasons why hops were initially put into beer hundreds of years ago, was because they noticed that, hey, when we put these in here, the beers don't turn sour. So let's use hops, or sour as quickly, right?
Yes, of course, but we have to remember that this is always combination of many factors.
Right.
Yes, in beer, we've got hops, we've got alcohol, but we also have low pH. And those three factors actually protect this liquid from the spoilage. And to be honest, it protects on some microorganisms, and on some or other microorganisms will still be able to spoil beer. It's not a holy grail. It's not 100% proof. So that's why we still have to pasteurize it. But if you don't pasteurize and you've got a beer that is high hops, the risk of spoilage is smaller. So it's sometimes I've got kind of slightly arguments with people who doing lots of research with hops that they say, oh, they are antimicrobials and stuff. I said, yes, they are antimicrobials, but to be honest, everything is antimicrobial. Water is antimicrobial for certain microorganisms. Oxygen is antimicrobial for another microorganism. Another microorganisms are affected by pH differently, and alcohol. So yes, there are lots of microorganisms that are affected by the hop extract, but there are lots of that are not affected. And also those microorganisms that are not affected, they evolve by many, many years around the beer environment, and that's why sometimes they survive.
Yeah, exactly. I'm reminded of an episode where I was talking with David Quain, who's the editor of the Journal of Institute of Brewing, and we were talking about non-alcoholic beers and how non-alcoholic beers, especially the ones that are lightly hopped, you've now changed those conditions, right? You've now removed the alcohol, which are toxic to some microbes. You've now reduced the isoalpha acids. You've reduced the hop content, that's antimicrobial, and you've oftentimes raised the pH because you're not, mashing for as long a time. There's not as much fermentation characteristics. There's not as much organic acid being produced by the yeast. So you've changed three of the primary characteristics in non-alcoholic beer, and that's really interesting, right? We think about pulling these different levers, and that's kind of what makes, I guess, pasteurization difficult. And one of the things you mentioned in the very first episode that we did, I think it was episode 71, was D and Z values are going to be dependent on the beer, right? On the beer that you're looking at.
Exactly, because it's depend on those factors.
Yeah.
More we test it, more we know about this. So at the moment, we don't have enough data, so we cannot clearly say, and we cannot clearly answer this. What's the combination? Also, in this study, we only look for the one kind of component, isomerized isoalpha, but how complex hops are. So, it may take us long time.
Yeah, to fully elucidate it. And that's, again, maybe we'll have you back on the show to talk about the next steps in the research. But you may also mention something, too, that some of these compounds, like you said, like oxygen is toxic to a lot of microorganisms, but then it's not toxic, obviously, to yeast. Yeast really likes oxygen. And so there are organisms that are sensitive to hop acids and others that are not sensitive to hop acids, right? And I think one of the things you mentioned in the paper is like lactobacillus and pediococcus especially are not as sensitive to hop acids as other bacteria. Now, they may still be sensitive, just not as sensitive as other bacteria, right?
Yes, that's a very interesting fact also, because if you would pick up a microorganism, lactobacillus or pediococcus, that is isolated from the food, they would not be as resistant as those that were isolated from the beer.
Yeah.
In fact, if you're looking for lactobacillus microorganisms in food, you're using completely different methodology. You grow them microaerophilically because they are microaerophilic, if those from the food in product. But in beer, we know that they are more likely strict anaerobe, that prefer no oxygen at all. So even those are the same kind of group, they will be different. Of course, this we know now that this is because there is specific genes that are hop resistant genes. So, and they were again developed. Those microorganisms developed those by many years, being in the environment, building their resistance.
Yeah. Yeah, and it's interesting too that like you said, the hops themselves have a bunch of different things, right? So the genes may develop a resistance to isoalpha acids, but hop pellets also have non-isomerized alpha acids, right? Alpha and beta acids, and then they may be isomerized or not, right?
Oh yes, and we can do research for another 200 years, yes, because always we will be finding some new, components and new.
Well, that's excellent news for you and me, because that I like the career stability there.
Well, I'm not sure if I want to explore this for all my life. I like, after a few years, I want to do something different.
Very true.
Yeah,
very, very true, right? Do something other than just looking at pasteurization and hop acids. But all right, so let's talk then a little bit about hop acids and how they interact with microbes. And again, we won't go too far into the science on this, but let's talk sort of generally about how do hop acids impact microbes?
So they interact similar, like kind of most of the essential oils. They would interact with the membrane of the cell membrane of microorganisms. They would interrupt it, and then it would make difficult for microorganisms, for the cells to actually function, to move kind of nutrient and everything what the cell needs through the membrane. So if this interacted, then slowly there is a lack of nutrients and then the cell die.
Yeah.
Of course, we know this is well explored in many papers, but nobody actually was looking into it. What's happened in combination with hop extract and heat resistance. So we only were looking into to find out what's the extent of this is. But we were not looking for exact mechanism, what exactly has happened.
Right.
You have to remember that we are not of course, we are academic, but we at the brewing side here, we are trying to look for the practical solution for the industry. And we want to leave this kind of strict academic question to the scientists at universities. So we just want to concentrate on part of our job that we believe we're doing the best.
Yeah, exactly. Right. The practical aspects of this for sure. And again, yeah, you're not looking in this research to try to understand the mechanism for hop acids, because there is a lot of research out there that's looked at that. And there's really interesting information, right? But the takeaway from here is that hop acids impact the membranes and they stop the cell from taking up nutrients, right?
Yes, exactly. This is kind of most basic, but accepted kind of explanation for this.
Okay. Yeah, exactly. And so then let's remind everybody too, we've been talking a little bit about bacteria, lacto and pediococcus, but again, the second episode, the second part of this, your second paper, was these microbes called yeast ascospores, right? And those are particularly heat tolerant. So let's remind everybody, what are yeast ascospores and why are they heat resistant?
So yeast ascospores would be germinated by yeast in the response to nitrogen starvation, in the presence of poor carbon source and presence of acetate. The ascospores are kind of a natural defense mechanism from the yeast to survive a very difficult condition in the environment, especially if the conditions are changing and the population wants to survive. So they transform itself to spores, and spores are very resistant. The metabolism is slowed down. The membrane is completely different, and it's proven that they can survive very harsh conditions. Not only heat, but also disinfectant, pH changes, all sort of desiccation, and they are much more resistant. And this is not only known for yeast. This is very common for bacteria. We've got with Clostridium species and Bacillus species. They do two species that produce spores, and they are super resistant. Not as much as yeast, but they are much more resistant than vegetative cells.
Yeah. Yeah, I like the way that you described this too. I mean, one of the big takeaways for me from the last episode is it's sort of like the yeast is protecting its DNA. It's putting its DNA into this capsule, like a time capsule. Like if you can imagine, putting something in like a, super thick steel so that it survives a nuclear holocaust. I mean, essentially that's what the yeast is trying to do, right?
Yes, and it's probing every so often. It's probing if the environment is changing and there is a favorable environment for growth. They would start to grow very rapidly. Yes, so yeah, as soon as there is a nutrients and there is no acetate and the environmental conditions are better, they would start to germinate.
Yeah. So you've got the nitrogen deficiency, and then as the environment changes and there's more nitrogen available, like for example in your packaged beer that's sitting on the shelf, those yeast ascospores come alive, and they, like you said, they start to grow rapidly. They make new yeast, and then we've got all kinds of trouble in our packaged beer. And so that was the second study you looked at. You were looking at the heat resistance of these different microorganisms to try to understand how tolerant are yeast ascospores. And in setting up your study, you set up a non-hopped beer, or not non-hopped, but a lightly hopped beer, and then a hoppier beer. And you noticed that there was a difference in the heat resistance between those two beers, which is really interesting. And so I think that's where we get into sort of the next natural step of the research was, has anyone ever looked into how yeast ascospores might be impacted by hop acids? I'm guessing no.
No, there was quite few scientists that they were explored heat resistant of yeast ascospores, but it was very strange that nobody actually looked into this. And our approach was very simple. We took a kind of American style lager, very low hoppy beer, like traditional, and kind of German style lager, which was 25 bitterness units. And we could see that massive difference, to be honest. It's shocking that nobody was looking into this.
Right. Yeah. So that's really interesting, and it sort of raises the question for me too. Is like, what about IPAs? Because you're talking about 25 bitterness units in a German, Pilsner style, but what if you're up at 50 or 60 bitterness units and you have higher isomerized alpha acids?
With those beers, it's questionable if you actually need pasteurization as well, because the composition of beer may be, self-protecting. But if you're removing alcohol, you may create a problem as well. And if I can come back to why maybe not so many people were investigating this, because to be honest, yeast ascospores we find out that they are not kind of easy to investigate. When we did try to produce ascospores, we seen that the variation of the basic characteristics each time we did it was different. So at the beginning we had some very strange results, but of course we learn with time, and now we're trying to understanding more why this happened. But it wasn't easy thing to do, and it's not much information. First, when we start looking into it, I as a food scientist, I wasn't even bothered about yeast ascospores, because for me it was all about pathogens and stuff. But when I explored yeast ascospores, I discovered new world.
Yeah, exactly. Right. I mean, you're worried about pathogens like Clostridium and all that sort of stuff in foods, right? Not ascospores. But so then I know this isn't, the topic of your research, but I do kind of wonder. So do you think, and maybe just if you don't mind speculating, and you don't have to, but do yeast ascospores, would they be impacted, similarly? I mean, is you mentioned that the membrane and like that's surrounding ascospores is different than like a normal yeast cell. Would you expect it to still be impacted by hop acids?
Yes, and we've proven this, that they are, but I don't know what mechanism it is. We know that, ascospores are much more resistant because the wall of the ascospores is built differently. Mm-hmm. Also, you actually have two walls, because one wall is around each ascospores, but then is also wall that is the ascus, so it's actually second wall. What else I can add to this? So I will have to leave this to scientists at universities to find out. But there's no mechanism like we know from the lactobacillus and stuff. There is no hop resistant gene or anything. I mean, we know that yeasts are happy to grow in the presence of hops. So we know there is nothing like this. But with heat, if you applied heat, there is basically maybe loosening this structure of ascospores, and then heat can penetrate much faster. So this is more kind of mechanical kind of mechanism.
Sure. Yeah, more mechanism study, and certainly an opportunity for people to study in the future. Like, what's the actual mechanism that's happening here? Because again, you've proven that that yeast are or that ascospores are impacted by hop acids. If no other reason than even your second study, right? I mean, we'll get into the results of this study, but your second study showed differences in the American style lager and the German style lager, very clear differences in their heat resistance. And this reminds me too, I was reminded of episode 7 of the Brew Lab way back, where I talked with Cameron Selna, and he was looking at actually conditioning beer serving lines with hop extracts prior to putting them in service, and it worked. He was noticing lower microbial load on beer service lines, but if you run some isomerized hop extracts through there, they don't have as high of microbial load after significant use. And so this is that kind of a similar concept, I guess, right? Like that these hop extracts are doing something that's reducing microbial stability.
Yes, but I think that applying kind of a coating on the tube is much easier, because you would apply much higher concentration.
Right. Yeah.
Mm-hmm. In beer, it's much less. You've got 50 ppms of iso extract acid, but in the tubes, in the pipes, you can apply much more. So it's probably a bit easier to achieve. But yeah, of course, there is lots of questions which. I mean, his study he did a lot of work. I did listen to his podcast, but, I just have to come back to it, because, he explored a lot of stuff. So I would be happy to come back to it.
Quite a bit, right? He explored a lot of stuff, and that's a very good point, right? I mean, he's talking about coating the inside of that, and there's a whole lot of questions even he mentioned too, right? That there flavor implications and all that sort of stuff. If you're putting, super concentrated isomerized extract, are those first several beers that you pull off going to be super bitter? So all kinds of questions to that too, which I understand that he's studying further, and that they're doing more research down there. Maybe we'll get him back on the show to do an update on that soon. But it is kind of interesting, right? So we are seeing, and again, this is supported a long time in the research. Like, we know that hops have this sort of impact on microbes, and especially bacteria, and that's one of the reasons why hops are in beer. And so it's pretty interesting. So the goals of this study, why don't you kind of walk us through what were the goals of this study? I'm guessing listeners can guess, but why don't we just go ahead and say it?
Yeah, so from this second study, we seen that there was an impact. There was the difference. That bitterness make a difference to heat resistance. But because we at the second study we are using two different beers, we wanted to make sure that actually is this impact associated with the bitterness compounds or not. So we needed to have the same kind of beer, contaminated with and adjusted with the one kind of a bitterness component, just to show that actually this component make a difference. So to do this, we got alcoholic and non-alcoholic light lager. The bitterness was, I think, about 6 or 8. And then we artificially added isomerized hop X alpha acids, and then just to bring it to IBU of 25 and 50. And then we did all the heat inactivation experiments, and this way we could show that, actually in this beer, because those beers are the same, alcoholic and non-alcoholic are the same, produced the same way, this only is impacted by the bitterness. Of course, this study was limited because we were looking only for one component. We actually, at the beginning of the study, we had 2 components to investigate, but we weren't able to even try the second one.
All right. Well, yeah. Well, I'm excited to hear more about the study and look at the results. And we will do that when we come back after this short break. One of the biggest improvements to my brewing practices was the upgrade to stainless steel, and Delta Brewing Systems offers some of the lowest prices on high-quality stainless gear, like the Firm Tank, which holds 8 gallons or 30 liters of wort, comes with a domed lid to reduce the chances of a messy blow-off, and it can hold up to 4 psi of pressure. Delta Brewing Systems also has their own line of brew kettles, as well as one of the lowest-priced all-in-one electric brew systems out there, and their prices are remarkably affordable. If you're in the market for legit stainless gear that won't break the bank, you've got to check out deltabrewingsystems.com. We all know that designing recipes is really fun, and doing it well is so much easier with good software. We at Brewlosophy recently made the switch to Brewfather, and honestly, y'all, we could not be happier. Brewfather utilizes the latest technology to bring you the most robust cloud-based recipe design software that can be accessed anywhere, on your phone, tablet, desktop, and even offline. It also works seamlessly with numerous third-party devices to make it easier to monitor every step of your brew. I know change can be difficult, but trust me when I say you need to go to brewfather.app today to try it out for yourself. That's brewfather.app. Hops are one of the main ingredients in beer, but historically it wasn't always this way. So hundreds of years ago, spices, herbs, and other aromatics were added to beer to offset the malty sweetness. And one oft-touted reason that hops were added to beer is their antimicrobial capability. So Greg, in the first part of the series, episode 71 of the Brew Lab, we mentioned that beer pasteurization methods hadn't been updated since, 1950 or 1960? That's incredibly surprising. We're in 2023 now. Well, in 2018, I think, when your first paper was written, right?
Yes, I mean, yeah. In 1951, there was a guy, Del Vecchio, who did the study, and all the guidance about pasteurization was based about his paper. He clearly said in his paper, please don't use it for the kind of general. But people ignore it and they use it. And then for later time, we had a different guidance. We got EBC, a manual practice of beer pasteurization from 1995, but which is very good guidance for the time that it was, because it was not, we didn't produce so many different beers, but because now we produce alcohol, non-alcoholic, different bitterness, so many different beers that this need to be updated. And this previous guidance didn't look into bitterness at all. Didn't include this, didn't include the bitterness, didn't include pH even. So we just trying to find out more about this.
Yeah, exactly. And that's what we did, right, in those first 2 episodes. And here we are in 2023, recording and publishing this episode. Well, I guess this research was published in 2022. So, we've been using hops in beer, and we sort of understand antimicrobial capacity of hops, but we're still learning that we don't know everything about pasteurization, right?
Yeah, I mean, very often I speak with the kind of, brewers, and, they are very skeptical. So, yes, and you're still doing pasteurization work. Like, come on, so yeah, it's neat, another 15-minute discussion to explain why we're doing this, so they get it. But most of kind of traditional brewers don't believe that actually there is much more needed. But to be honest, time is changing, and then pasteurizers quality of pasteurizers are improving. You can be more precise, especially with the sustainability, which is talked everywhere. We have to try preserve energy everywhere, but also time. If you can reduce each pasteurization of each batch by 15 minutes, that means that you can produce more in a day. Or you can do it at lower temperature, and the product maybe taste better. So there is still to do, but some with the kind of hardcore brewers, we've got sometimes arguments.
Well, sure, right, exactly. And that is right. I mean, one of the original reasons why you wanted to look into this is the flavor impact on beer. If you're pasteurizing things for a long time at high heat, there could be potential flavor impacts. And so again, to brewers that are looking to pasteurize, the whole purpose of Greg's research is to give brewers guidelines, and the Campden BRI research is to give brewers guidelines so that they can take that and use that in their brewery and, have their the pasteurization requirements, perfect for their specific beer. Right, Greg?
Yes, more we do, more we can learn, and we can apply this much easier. So at the moment, the brewers can buy this specific microorganisms in their lab. They can produce ascospores. They can do their own study, and then they do whatever they want with this. But because at the moment, lots of brewers don't even challenge pasteurization. Right. Mm-hmm. The way how we challenge, we actually inoculate beers with the high level of microorganisms, and we set this through the pasteurizer at the end, and we check if this was pasteurized. In the brewery, people would not do it because it was either too risky, or so they would rather run the current system and apply more pasteurization units that is actually required. But but very often we know that sometimes there may be failure with pasteurization. So even if your target of pasteurization is 15 PU and something went wrong and went in 10 PU, mm-hmm. You've got a dilemma. Should you do this batch again, or this batch is still okay? Yes, if you've got data for this to show that it's still okay, you can release it. You have assurance that it's not going to spoil because you kill 1 million of most heat-resistant ascospores per meal.
Right, which is good, exactly, and that's what brewers are looking for, to make sure that the pasteurization is actually working and destroying the microbes in their beer. And so we need to talk about hop extracts. All right, so that's this research project that we're studying today. And you mentioned before the break that you did beers, right? You sourced 2 American lagers, so very lightly bittered. One was 4.5 ABV, and the other one was non-alcoholic, so 0.0 ABV. So I want to ask one of those questions: why the difference? Why look at a non-alcohol and a normal?
Well, we wanted to see what's happening when the alcohol is present and when there is no alcohol as well. So especially with current, lots of beers, non-alcoholic beers, we want to validate this for non-alcoholic beers as well. And what was important, that those beers were coming from the one manufacturer, that they would be prepared similar way. Mm. And they would have a low bitterness. Yeah.
And you mentioned that you spiked it with an isomerized hop extract. That was Isolone from Calsec Europe. That's an isomerized hop extract that contains between 28.5 and 31.5 isoalpha acids. And you use that to spike each of the beers. So you've got, again, 2 American lagers, a 4.5% ABV and a 0% ABV. And you spiked those to 25 or 50 IBUs. And again, the control also had no spike, but it would have a low bitterness because the beers themselves had like 8 or 6 or 8 or something like that IBUs, right?
Yes. So at the end, we would do the study at 3 points of bitterness. Mm-hmm. Initial, which is about 8 or 6, 7, 25, and 50.
Yeah. Okay, perfect. Yeah. So then you've got, if we're looking at bitterness, this is set up great. And what 2 microorganisms did you decide to use?
Well, we use our biological indicator that we validated, which was Saccharomyces cerevisiae. So it was ascospores. So this was validated. It was picked up from the 30 from the second study as a most heat resistant. And also we use, as we always use in those kind of study, representative of spoilage bacteria, which was Lactobacillus brevis, which we found in the previous study that was most heat resistant bacteria as well.
Right.
Of course, we knew that heat resistant of yeast ascospores is much higher than bacteria, but always we wanna have reference to bacteria, right. How much more heat resistant it is.
Yeah, how much more resistant it is than the bacteria. Yeah, and that's cool, right? I mean, again, you're building on your two previous research works. So the first study, you found that Lactobacillus brevis was the most heat resistant bacteria, and the second study, yeast ascospores are more heat resistant than Lactobacillus brevis. And so in this study, the third study, now you're able to compare and say, okay, how much more heat resistant is ascospores compared to Lactobacillus brevis when there's bitterness applied, higher levels of bitterness.
Yes, and we always looking for new organisms. So we're looking for something that maybe we are very confident that Saccharomyces cerevisiae is most heat resistant and produce good ascospores, but we may be looking for some replacement for Lactobacillus brevis. We believe that maybe there is something more heat resistant, but it will not impact our study because it will be still much less resistant.
Right, still much less resistant than the ascospores. So of course, then you have to build up the ascospores and the Lactobacillus brevis, and I'm assuming you spike those directly into the beer.
Well, first we're doing D and Z values, so we're doing this separately. Yes, so so what we have to measure? We have to measure D value, two kind of parameters for heat resistance, which is D value and Z value. D value is decimal reduction time. This is time in minutes that will, at a certain temperature, that will reduce population by 1 log. So, for example, time is required to reduce population from 10 to 1 or from 100 to 10.
Mm-hmm. Yeah.
So this is one parameter. So we have to do this at a number of temperatures. So we do it at four different temperatures. And when we have these 4 temperatures, we use those 4 D values to put this to the graph. And from the graph, we can calculate Z value. And Z value is defined as a change in temperature required for 10-fold change in D value. Sorry, this may be not straightforward to follow, but for example, if Z value is 5 degrees, that means if we increase process temperature by 5 degrees, D value will reduce by the 1-fold. So D value would be reducing from 10 to 1. So how are we doing those experiments? Those experiments we're doing in capillary tubes. Why are we doing in capillary tubes? Because the capillary tubes, they are very thin. They made from the thin glass. Diameter is about 1 millimeter. And the heat penetration is immediate. I mean, in fact, we measured this. We've proven that it takes 6-7 seconds to warm up entire liquid in the capillary tubes to target temperature. So how we do it? So we have those capillary tubes, and into each capillary tubes we are adding our mixture of our product, our beer, with yeast ascospores or lactobacillus. So we inject 50 microliters. We seal ends with the heat, and this is our sample. So if we've got 3 of them, we can put them into the water bath, set at target temperature, and hold at the specific time, and enumerate microorganisms before and after heat treatment. And we doing for each temperature, we would do this several times. Ideally, we want to do it 8 or 9 times at different temperature. So our kind of standard is that we want to have 5 valid time points with entire reduction of microorganisms. So if we inoculated 1 million microorganisms, we want to see at the last tubes nothing.
Mm-hmm. Yeah, it makes sense. And so then what temperatures did you use for doing these, the D and Z values?
So for alcoholic beers, we had to use a slightly lower temperature because the highest temperature was 64, and for a non-alcoholic was 65. And because the D values were different. So for alcoholic beers, we did 64, 62, 60, and 58. And non-alcoholic, we did 60, 62, 64, and 60 degrees. Sorry, you might have to translate this to Fahrenheit.
Yeah, that's okay. Yeah, no problem. I can translate to Fahrenheit. So the high temperatures were for the alcoholic one was 147, and for the lowest for the non-alcoholic was 149. So he did temperatures at 136, 140, 144, and 147 for the alcoholic beer, and then 140. 144, 147, and 149 for the no-alcohol beer. So these temperatures were a little bit higher for the no-alcohol beer to see the reduction in the microbes. So is that the alcohol effect on the microorganisms?
Yes, I mean we wanna. Always we want to do temperature, the trial at highest temperature for the specific product. So because heat resistance in alcoholic beers is lower, we have to do it at lower temperature. Our target for the doing inactivation trials is doing intervals every 10 seconds.
Yeah. Okay.
So the highest temperature which we could use 10 seconds intervals for premium alcoholic beer was 64, and for non-alcoholic was 65, basically.
Okay. Yeah, it makes sense. All right. Well, then I think let's start with some of the results. So I think we should start with sort of the big takeaway. So did isomerized hop extracts have an impact?
Yes. As we seen in previous study, we explored this further. And for example, if we're talking about non-alcoholic beers, if we look only at the D60 value, which is decimal reduction time at 60 degrees, we had D60 was 12 minutes in an initial beer, which was 8.6 IBU. At 25 was 8.6, and at 50 was 6.
Wow.
So at 50, it was twice less.
Wow.
Yes.
Yeah. Right. So twice the time difference, right? 12 minutes for a log reduction with the unspiked beer, but then at 50 IBUs, it's half as much. It's 6 minutes.
Yes. So it was basically you can transfer it as it would require twice less heat during pasteurization, basically, if you want to kill it. So this was quite powerful, and we recalculated this for how much pasteurization units would be required. So for this beer, non-alcoholic, at initial pasteurization, at initial bitterness, we would need 80 pasteurization units. But if the beer is at 50 PU, you would only need 40.
Wow. Okay.
Of course, now it's easy to say, oh, yes, we would need less. But, if you haven't done the study, you would question, is it 40 or maybe 20 or maybe 10 or maybe 60, or just to be easy, let's do it the same. Yeah. So most of the people would do the same, or 80 PU, or maybe slightly less, 60. But now you know that it's 40.
Well, that's interesting. So you did, again, you did like a 25 versus 50, and that's roughly double. And then when we look at the pasteurization units, there's also sort of roughly double, right? I mean, 25 takes twice as long, or twice as many pasteurization units, twice as long for the D value as the 50 IBU. So is it like a linear relationship between these?
I think this is different for different beers.
Yeah.
So also, we would have to test specific beer, which are actually properly brewed. Not that you adding, iso extract to it, So this beer would have much more hops components. So it would be even greater impact than only with one component, I believe.
I see. Yeah, I see. Interesting. Yeah. So it's because of all of the other different pieces of the beer, right? The pH, the alcohol, the different alpha and beta acids concentration, all the other stuff that's in the beer. It might change that relationship.
Of course, also we would take to account pH as well. So yes.
Yeah, good one. I always seem to forget pH. I think every time that we've done the show, I've always seemed to forget pH. I sort of take it for granted in beer, but it's not. It changes quite a bit. And small changes in pH can have big impact, which is also important too. Okay, so all right, so we see the change in D and Z values, and the what you were mentioning, the D60 of 12 minutes being reduced to 6 minutes at the highest IBU. Was that for the ascospores or was that for the bacteria?
It was for ascospores.
Okay, wow, yeah. So that's the most heat resistant, right? So the most heat resistant bacteria are seeing a really big change. But what about the bacteria? Did you see similar results?
Well, because we did it only at one temperature,
right? And
generally lactobacillus is not very heat resistant. We haven't seen great difference, So I can tell you what the differences were in non-alcoholic. At initial IBU, it was 0. It was 1 minute. Yes.
Okay. Yeah.
At 25 was 0.76. And at 50 IBU was 0.6.
Okay.
So, you can speculate, is it, it is significant because we check this.
Yep.
But, if you look at this data like this, it doesn't seem to be difference. But, if you would do this tries at 55 degrees, you would see how different it is.
Right. At a lower temperature, right, where lactobacillus is less resistant.
Yes, but in this for the lactobacillus, we just wanted to show that more, that is much more less, but actually it shows us that there is impact as well of bitterness as well.
Exactly. Yeah. So like the practical impact of this for lactobacillus is relatively minimal, right? 1 minute versus 20 seconds less or 30 seconds less is not going to change much practical impact. But you can say that statistically, if you look at the differences, there is definitely a difference, even in the bacteria, in terms of the heat resistance. And of course, if you had changed the temperature, you would see that those seconds get spread out to minutes, and it would be much easier to sort of see the difference at lower temperatures.
Yes, but because most of the time pasteurization done at 60 degrees, this is what is the best to show it at 60 degrees.
Yeah.
This is, one between 0.6. Okay, nobody will be worried about those kind of seconds.
Exactly, right? Nobody's going to change or worry about the time savings
Yes.
for the extra 20 seconds. But the ascospores is very interesting, right? 6 minutes less, is 6 minutes less that you're under 60 degrees Celsius, right? That the beer is exposed to that temperature, and that can have significant changes, like we talked about, to flavor or to aroma or to other things that are going on in the beer. And so this is really interesting. So you have proven that IBUs, or that bitterness, the isoalpha acids, impacts the D and Z values, and it's pretty cool to think about that. That's sort of a double relationship, right? At least with respect to the ascospores, right? 80 PUs versus 40 PUs to get rid of the ascospores in these beers at the alcohol levels.
Yes, this is very powerful because it's give you right value, and you can adjust your process to this, and you can save lots of time and money.
Yeah, exactly. Save a lot of time and money, and not expose your beer to those high temperatures for a longer time. You may have mentioned it and I may have missed it. Did we what about the difference between the premium lager and the alcohol free lager? Did we notice big differences in D and Z values there?
Well, of course, the D values for alcoholic lager were much lower because of the alcohol.
Yeah.
But we observe a similar trend.
Okay.
But impact in alcoholic beers was not twice lower, was maybe 1.5 lower. And so reduction from D60 to at initial bitterness to at 50 was 1 point less, 1.5. But Z value are interested because in alcoholic beers, Z values are slightly higher, and in non-alcoholic slightly lower, which we observe this in many cases, that if you've got more challenging environment for yeasts. Of course, D values will be lower, but this may be compensated by the Z values being slightly higher.
Okay, interesting.
Yeah. Of course, in this study, we don't show this that much because, for example, I show you a non-alcoholic lager at 25 IBU 60, we've got Z value of 4, and in alcoholic, at the same condition, we've got Z value of 4.5. So it's only 4.5 degree. So it's not much, but there is a trend. So something we've seen this that is quite common.
Interesting.
But it's very difficult to build up anything from this because this was for a specific beer, and in different beers, different factors may impact this. So we quite resistant to make any kind of assumptions that would translate this.
Right to other beers, right? I mean, this is a premium lager that's spiked with a isomerized hop extract to show the impact that bitterness has on ascospores and lactobacillus heat resistance, right? Again, you're not looking at a hazy IPA, for example, or a bitter IPA, a West Coast American style IPA, or like an English porter or something, right? Which might be, very different or have very different resistance characteristics.
Well, we would love to do it, but of course, there is a limit of the funds available for this.
Yeah.
But yeah, well, I mean, we will do it. I mean, my colleague is doing a PhD in this area, so he's building a model that he will put bitterness into the model, pH and alcohol, and this model will give him a D value that it should be applied, and he could calculate pasteurization. And based on the model, he will check different beers that are brewed properly, how far away is this. So we will be doing more work in this.
Awesome. Yeah, well, I'm really looking forward to that research as well. We'll have to have him on the show to talk about that once he's got it. So some pretty high level takeaways then. Yeah, I mean the isomerized alpha extracts, at least, or bitterness, seems to have a big impact on ascospores heat resistance and bacteria too, for that matter. Although again, the bacteria is much less heat resistant than ascospores. So the question then, I guess, future research, what's next for you? Or what are you going to look at next in terms of pasteurization? Or are you done with pasteurization for a little bit?
Well, at the moment, I pass this to my colleague to explore, to do his PhD. So he's looking into creating this model. So at Campden BRI, we purchase a new kind of pilot plant pasteurizer that we can, use more, do this kind of trials more precise as well, because we're doing Z and D and Z value on the lab, and then we're moving to industrial pasteurizer in pilot plant. Also, my colleague PhD, looking into how the shelf life of ascospores, for example. Yeah, if we prepare the ascospores, how long we can use this for experiments? Should we use them straight away, or are they changing? Do they change heat resistance? He was also looking into what actually is the best agar to recover yeast ascospores after a heat shock. At the moment, in all these past studies, we were using yeast and malt agar, but he explored lots of different agars, and he found that there is a slightly better agar for this. So, there is a lot to explore, and also, with this is first indicator, biological indicator that heat resistant is lower than the resistant of bacterial spores, so it can be used for lots of other products as well. So we can use this for non-alcoholic spirits. We can use this on seltzers. We can use this basically on any product that the yeasts are main spoilage factor, because we know that this is most heat resistant. So we're doing lots of work on huge number of products. We were trying wines as well. Yeah, we're doing a lot. Anything that clients come to us, we can help.
Wow. All right. So there's a wide range of products and stuff that this research can help as well. And before I forget, I wanted to ask, what was the name of your colleague? We should probably mention his name.
Oh, yes. This is Chris Rowley.
Okay, yeah, perfect. Good. Well, I look forward to Chris's research and to having him on the podcast whenever he's got that research ready to go. Well, I think we covered sort of the biggest takeaway for today is that again, isomerized hop acids seem to reduce ascospore and bacterial heat resistance. But I do always ask this question towards the end of the show, Greg, and if you want brewers to take away just one thing from today's episode, what would it be?
Well, the message to kind of people who run pasteurization or the quality managers, just try to perform validations, check what the D and Z values are for the specific. You can buy this yeast from the national collections, or just you can come to us for help. And believe in data and run validation in pasteurization and adjust pasteurization, save energy and time.
Yeah, save energy, time, and hopefully it maybe improve the flavor of your beer too as well.
Exactly. Yes. Yeah.
Yeah. Well, is there anything else that you wanted to talk about that we didn't get to today?
Oh no, thank you very much, Cade. I think we cover a lot. At the beginning I wasn't sure if we will cover this for whole hour, but actually we managed to do it. So we filled for 1 hour. This was our shorter research, but actually we managed to do this. So
we did.
Thank you.
It was a shorter research, but really interesting takeaways too, right? And so I'm glad, and I'm glad that you joined me on this. I'm glad that we also were able to tell this great story about pasteurization, looking at the first 2 research projects that you did as well, and then sort of tying it all together here in isomerized extracts. And of course, I'm now excited for the research that's coming in the future. So thanks again, Greg, for joining me in the Brew Lab.
Thank you very much for having me. Thank you.
All right. Well, listeners, in the show notes, you'll find a link to Dr. Rachon's paper, which is titled The Impact of Isomerized Hop Extract on the Heat Resistance of Yeast Ascospores and Lactobacillus brevis in Premium and Alcohol-Free Lager, which was published in the Journal of the Institute of Brewing and Distilling in 2022. So be sure to check out all of the fun stuff we're up to at brewlosophy.com. And don't forget to listen to our other show, the Brewlosophy Podcast. The Brew Lab is a production of Brewlosophy, where they who drink beer think beer. Don't forget to visit brewlosophy.com to read about our weekly experiments and other brewing adventures, and listen to us talk about it on our other show, the Brewlosophy Podcast. Thanks to all of our sponsors and patrons that help make this show possible. If you'd like to receive a reward for helping us do what we do, visit patreon.com/brewlosophy to see how you can do just that. Thanks so much for listening. We'll be back in the Brew Lab with another guest next week. Until then, think beer.